Hello. Can you hear me? You're muted.
Yes.
Okay.
I think, are you in the lab?
Yeah, I'm in the lab. Is it too echoey?
There is a little bit echo there.
I can try opening the door. How does it still have like a little bit of an echo here?
I think it's okay.
Yeah, I think it sounds better. Just give me a few more minutes.
Do you want to put your camera on?
Yes. One second.
Also, I'm not the host. Is there a way you can make me the host?
Yes, let me see. This is the first time I'm hosting a Zoom meeting. Okay, I can make you. You want to be the host or the co-host?
A co-host. I think there is a point of time where you will have to exit the room.
Okay.
So I have to be the host or co-host to maintain the room.
Right. Okay.
And then you can put your PowerPoint on, I guess. Is it on?
I'm looking for it. Okay, I found it.
You want to probably start with your, you get the PowerPoint started and then you share the screen that's actually present. Okay.
Like that?
Yeah, that should be good.
Okay. Give me a second.
So I think your committee members are here. Dr. Park and Dr. Choi, can you confirm that you're here?
Hello, everyone.
Okay, good. So we can get started. I think we'll start with an introduction. So,
well, let me. Okay.
Yeah, so Miguel Gonzalez has been in my lab for over a year, and he was funded by a UCF seed grant to conduct research regarding how children would be able to control their muscles to control as a way to control external devices. And it's been a tough year, and especially for human research. And I'm glad that we actually got enough subject in the last few months. Miguel spent a lot of weekends and after hours to collect data with children's subject because their parents has to work during the day, so we usually just schedule during off hours. So it was a lot of effort to work with children subject. And so without further ado, I'll let Miguel to present his master.
Okay, thank you very much, Dr. Fu. Like you said, my name is Miguel Gonzalez. I am a master's student in biomedical engineering, thesis candidate. And the title of my thesis is Understanding Forearm Muscle Coordination in Children. So we'll go ahead and get started with the introduction and background for this. So the loss of a limb can have a dramatic effect on the quality of life that an individual experiences. Causes of amputation can range from traumatic injury or congenital causes. A prosthetic device is often used to restore some level of functionality to the user. The first upper limb prosthetic devices use simplistic mechatronic methods such as body operation, like movement of the shoulder to actuate the device. However, these early prototypes were often sluggish and required advanced motion planning, which added more cumbersome to the use of the device. So a successful prosthetic device is one that the user will actually use, and oftentimes the amputee will forego the use of the device simply because it's just too much trouble to actually use. Luckily, there have been many advancements in prosthetic device research that have led to some improvement in functionality. Since the late 1960s, researchers have been using electromyography or EMG signals as control inputs for these devices. By using surface electrodes placed on antagonistic pairs of muscles in the residual limb to measure the magnitude of the muscle activity. Simple movements are able to be generated, such as opening and closing of a prosthetic hand, or extension and flexion of wrist, etc. These advanced control methods have been developed in order to allow for a higher degree of freedom and functionality. One common advanced control method uses pattern recognition to classify EMG signals into distinct movement classes, which drive the prosthetic device. This method has shown a lot of success with high classification accuracies being achieved in adult subjects. There are some drawbacks and assumptions regarding this method, which will be discussed further. In contrast, most prosthetic devices meant for pediatric amputees are very simplistic due to considerations of factors such as cost and weight, as pediatric amputees must often resize their device to fit their growing bodies. So most research done in this field has solely been focused on adult subjects with fully developed sensio-motor control capabilities. And there exists very little data to show how children would perform using the more advanced control methods for prosthetic devices due to their developing neuromuscular structures and rapid growth. Additionally, it'd be beneficial to learn how to optimally design pediatric upper limb prosthetics and designing rehabilitation programs, as studies have shown that early adaptation of prosthetic devices leads to higher acceptance rates. The work done for this thesis attempts to address the gap in this knowledge and aims to provide a foundation for future clinical studies involving pediatric amputees. The main objective of this research is to validate and benchmark an experimental protocol that quantifies the ability to coordinate forearm muscle contraction in able-bodied children across adolescent ages. This will be achieved by conducting two different experiments aimed at evaluating the subject's performance in operating two different types of myoelectric prosthetic control methods. The one being the pattern recognition mentioned earlier, and the second being simultaneous proportional control. Okay, so next we'll go into a little bit of the necessary literature review to kind of understand the research involved. So under the muscle synergy framework, muscle synergy is defined as the activation of a group of muscles that contribute to a particular movement. This framework is very helpful in helping us understand how the central nervous system actually controls and produces our own movements by reducing the dimensionality of control from reducing by focusing on these groups of muscles instead of individual muscle activations or even the thousands of individual motor units that comprise each muscle. And there's been a lot of research done examining this muscle synergy framework. Typically, the way it is, the typical approach to measure, to quantify these synergies is to measure EMGs from a large number of muscles while performing some sort of complex behavior. The second part is to use computational analysis, such as non-negative matrix factorization or some type of independent component analysis or like PCA in order to identify the set of synergies from the recorded EMGs. Thirdly, it's necessary to evaluate whether the observed EMGs can be well described by the combination of these synergies. Usually it's a linear combination of these synergies. And fourthly, it's necessary to relate the identified muscle synergies to task relevant variables. And there are two types of synergies. There's the synchronous and time varying, which basically just means that in time varying synergies, the muscles can, there's differences in time between activation, while synchronous is the one that we're going to be looking at for this particular study, and basically just means that they happen at the same time. And also relevant to know is that the average of able-bodied adults, studies have shown that they're able to produce about 6 synergies. And as mentioned earlier, so one of the factorization methods used to calculate muscle synergies is non-negative matrix factorization, which is basically just a set of algorithms that are able to decompose an EMG matrix into 2 separate matrix factors, basically, with the non-negative constraint. So the non-negativity constraint is very useful, and it allows for this algorithm to be used in a wide variety of applications, such as facial recognition, but most importantly for this study is muscle synergy analysis. So one way that in MATLAB, the NMF algorithm is shown here. It's important to note that factorization is not exact. The W times H is a lower rank approximation to the non-negative input matrix V. And the factors W and H are minimize the root mean square residual between V and the W times H. Factorization uses a iterative algorithm that starts with random initial values for W and H. And because the root mean square residual might have a local minima, repeated factorizations might yield different W's and H, and sometimes it's necessary to repeat the process in order to make sure that the algorithm does not converge to that minimum. So there are many different types of classification algorithms that are used for pattern recognition, but they all follow like the similar common stages of signal processing, which include data pre-processing, data windowing, feature extraction, and classification. Pre-processing is done usually to filter out any background noise, usually with a Butterworth filter. And also the EMG signals are rectified. The data is then generally partitioned into these window frames consisting of smaller sample sizes. There is a balance that a larger window size contains more samples, which means that it represents more of the variation within the data, and therefore leads to higher classification accuracy. However, larger window sizes also take longer to compute. So there's a necessary balance to strike between window size, classification accuracy, and speed if you're looking for real-time applications. And a common window size is between 200 and 400 milliseconds. The next step in is feature extraction, which basically is some type of characteristic of the signal that condenses the information possessed within that signal and is used to discriminate between classes. So here is a table of some of the common features used for PR pattern recognition algorithms. All of these features have been tested extensively for prosthetic control, and generally and even combinations of these features have led to very high classification accuracies. For simplicity, we ended up using the root mean square is defined there because it's very, the difference in classification accuracies wasn't too great, and it also was the most, it was very simple to extract. And lastly, so once the data has all been processed and the feature extraction has taken place, the next step is to feed that into the classification algorithm. And two of the most common classifiers are linear discriminant analysis and support vector machines. LDA is a popular choice for data classification and dimensionality reduction due to its computational simplicity. This method works by finding a linear combination of features that separate the given data into two or more classes. It's pretty similar to principal component analysis, which also seeks to explain variance in the data. However, the biggest distinction being that PCA doesn't take into consideration the differences between classes. Support vector machines is another method that's widely used. This method uses the data points that are furthest away from the center of the class. So kind of like the extreme cases of a particular class in order to create these so-called soft margins, which are then used to classify new data points. And so studies have showed that both methods can achieve very high accuracies. However, SVM generally outperforms LDA, which is why we use SVM for this experiment. It's important to note that the PR control systems are often evaluated by their offline classification accuracies. However, studies have shown that there can exist significant discrepancies between offline performance and real-time performance. Nonetheless, offline classification accuracy is still a very useful metric that we will be using to compare and contrast adult and child performance. So that was for proportional control. And then so to add to the, one of the drawbacks of the proportional control, proportional control works by classifying the data into a distinct class. And so, and there is no in-between classes. Generally, if the prosthetic device would move at a fixed speed. So in order to kind of alleviate that problem and add more functionality, there have been other control methods that have been developed that take into consideration the proportional, the magnitude of the force being used to generate that movement. And that's where simultaneous and proportional control comes in. And here we discuss Fitt's law, which is a commonly used, I guess you can say paradigm that is used to evaluate target acquisition. So basically Fitt's law is a predictive model for human performance, commonly used to evaluate simultaneous proportional control systems. Fitt's law states that the amount of time required for a person to move a pointer to a target area is a function of the distance to the target divided by the size of the target. And so here we have this metric known as the index of performance or ID. There's a typo. The left one here, that's supposed to be, okay, sorry. No, that's right. I got confused for a second. So the index of performance is IP here, which is the index of difficulty divided by the movement time. And index of difficulty is defined as the log base 2 of twice the distance divided by the width of the target. And this basically means that the farther away, the longer the distance the target is and the smaller the target size, the longer it will take to reach that target. During these tasks, each movement can be split into 2 phases. There's the initial movement, which is a fast but imprecise movement towards the target, and then the final movement, which is slower but more precise movement to acquire the target. And we redefine it as first touch and basically the adjustment time. And the fifth law is used because it allows us to quantify and compare between different target distances. Okay, and lastly, so this is here's a quick review of neuromuscular control in children. It's important to note the differences in the neuromuscular structure between children and adults, as children are still in a stage of rapid development and have lots of physiological changes that adds to the complexity of studying pediatric myoelectric control. Several studies have aimed to quantify children's performance in motor tasks using a standardized test known as the Zurich Neuromotor Assessment. This test was used to assess differences in performance in children between several factors such as gender, age, and handedness. A lot of the studies that use this test have found that younger children perform slower than other children in motor tasks, and that there was a high variability within age groups. Additionally, the complexity of the task correlated with an increase in the age in which performance for that task reached a plateau or maturity. Yet other studies have indicated that several neuromotor capabilities are not yet fully developed at the time of adolescence, and that different capabilities mature at different ages. The other concern is that children have smaller forearms. So there is an increase in EMG in between the EMG sensors, which is called EMG crosstalk. Basically, that EMG crosstalk occurs when a sensor picks up electrical activity from a different muscle. So it can be difficult to kind of isolate exactly which muscles are contributing. Okay, so now moving on to methodology. Here is a breakdown of the subject characteristics for each experiment. 8 healthy adults and 9 healthy children successfully completed the first experimental procedure. And for the second experiment, oh, that's another step. Table 3 shows the subject's summary for experiment 2, which is a typo there. But there was also 8 healthy adults and 13 children. However, 2 children, subjects 6 and 8, were not able to successfully complete the procedure, and the resulting data was excluded from analysis. Subjects were recruited locally from nearby elementary and middle schools, and all human research was approved by the University of Central Florida, IRB. So, for the equipment, we used 8 surface EMG electrodes that were equally spaced around the forearm of the subject's dominant hand. These sensors communicate with the base station showed in figure 2 via Bluetooth, and that base station is plugged into the computer via USB. The EMG signals were sampled at 2 kilohertz. By placing the sensors equally spaced around the forearm, this is known as the dense sampling strategy, and it doesn't target specific muscles as opposed to the anatomical positioning strategy, which aims to actually place the electrodes above forearm muscles, specific forearm muscles. This method is not common for evaluating muscle synergies. However, it does mimic the typical electrode placement used for prosthetic device control. And it also aims to characterize it's useful for characterizing the muscle activation pattern without a priori knowledge of muscle locations. This is important given the differences in limb reduction among amputees and the differences in forearm circumference between adults and children. And this method of electrode placement is also commonly used for use with simultaneous and proportional controllers. Figure 4 shows the orientation of the polar plot. So during, so on the typical right hand, during the experiment, the rest position is shown in figure 4, as long as the orientation where flexion and extension corresponds with the 0 and 80 degrees respectively. Similarly, abduction and adduction correspond with 90 and 270 degrees respectively. Because electrode placement was mirrored between right hand and left-handed subjects, this allowed for the generalization in using these polar plots that will be later used to show the muscle synergies. Okay, so for the experimental protocol for the first experiment, this experimental protocol was based on several studies that utilized the NinoPro database, which is open source, publicly available database of EMG signals. So for this experiment, subjects were asked to mimic 16 different hand wrist movements, which were shown on a computer screen. Each movement was repeated 8 times, and each repetition lasted 5 seconds each, with 2 seconds of rest in between repetitions. Before each movement, subjects were briefly coached on how to perform each movement sufficiently. And subjects, especially children, were allowed breaks after several completed movements as needed in order to alleviate muscle fatigue. The movements are summarized here in this table and were selected from a list of commonly investigated movements, as explained earlier from the Nina Pro database. So for the data analysis, after the EMG data was collected, the raw EMG signals from each movement repetition were processed using MATLAB. The EMG signal is zero meaned and rectified, and then passed through a zero phase digital filter using the filter function and in MATLAB, and the transfer function coefficients of a fourth order Butterworth filter with normalized cutoff frequency of 0.01 hertz. In order to decrease variability in the signal and increase classification accuracy of the algorithm, portions of the EMG signal associated with muscle contraction, rest, and were removed. Additionally, data samples that lied above 3 standard deviations from the mean were removed. The remaining EMG data was then partitioned further into windows consisting of 80 samples each, or about 250 millisecond windows. And the RMS feature was calculated for each window. So performance in this experiment was characterized by the number of highly independent movements produced, which is based off another study where it was defined as a subset of movements with greater than 90% accuracy. So data from 5 movement repetitions shown there were used to create the training features for the SVM model, while the remaining data was used for testing. In MATLAB, the SVM model is based off of a learned template with a Gaussian kernel function and standardization set to true. The standardization is used to center and scale each column of the input data by the column mean and standard deviation. After training and testing, a confusion matrix was generated with all the movements and which basically showed their error rates or classification accuracy. The movement with the lowest accuracy was removed from the training and testing data sets. And this process was, and the SVM model was retrained with the data from the remaining movements. This process was repeated until all remaining movements had a minimum classification accuracy of 90%, which is how we are defining the highly independent movements. The number of highly independent movements gives an idea of the number of movements that could be generated using a prosthetic hand for that specific user with minimum training. And it's important to note, like I said earlier, that it's an estimation, as offline performance has been found to have some differences with online performance. The second part of the data analysis is the dimensionality analysis, where we try to find the muscle synergies. Data was further downsampled to 100 hertz. After removing rest periods, the remaining signals were concatenated across all trials and movements and normalized to have unit variance. We used the NMF algorithm to extract time invariant muscle synergies and their dependent activation coefficients from the EMG data. Much of the data analysis was derived from previous studies that extracted muscle synergies. The ultimate goal of the NMF is to capture the major EMG channel covariation patterns, thereby yielding an estimation of the dimensionality of the EMG data. To identify the number of synergies for each subject, we first divided the EMG data set randomly into two subsets, one for extraction and validation at a 75 25% split. The NMF was first computed using the extraction subset with a given synergy number K, where K ranged from 1 to 6. And the resulting synergy matrix was used to obtain H using the validation subset. Then both global and local variance accounted for, which is VAF. After the NMF were obtained from the reconstruction of the validation subsets. Again, as mentioned earlier, since the NMF algorithm may converge to local minima, the synergy extraction was repeated 20 times for each synergy number with a random subset sampling and random initial estimates of W and H. The result corresponding with a maximum global VAF for the given synergy number was selected. This entire procedure was performed with varying number of synergies from 1 to 6, or up to the number of electrodes, which in this case is 8. And basically the number of synergies for each subject was defined as the minimum K that would achieve a global variance of 95% and a local variance, local VAF, sorry, of 85% for each EMG channel. So,
I'm sorry, Mika, you want to speed up a little bit. We're 40 minutes in already.
Oh, okay. Yeah. All right. So for experimental protocol 2, the task was target acquisition. Basically, subjects were tasked with trying to reach a target as quickly as possible by controlling the cursor using extension and flexion of the wrist. There were 20 targets in each run and 18 runs in the whole experiment. Targets were evenly distributed between the left and right side of the slider. Each target had a fixed width. In this experiment, we examined the effect that the gain level had on subjects' performance, such as time to target. There were 3 different gain levels, low, medium, high. Each subject had one of 2 sequential gain orders, low, medium, high, or low, high, medium, low. In the second phase, the order was reversed. This process was done to reduce bias in performance due to the gain order during data analysis. If the subject did not reach the target within the time limit of 10 seconds, the movement was counted as a failure and the subject continued on to the next target. There was an initial calibration stage, which was used to collect 15 seconds of EMG data while the subject repeatedly performed extension and flexion of the wrist. Subjects were instructed to perform their maximum voluntary contraction force during the first 2 repetitions. This MVC was calculated for both directions by taking the mean of the largest 50 data points. And then the calculation of MVC is important in order to normalize the force required to move the cursor to the individual. That way, it can be a more comparable measure between subjects, as children will have generally, due to their smaller muscle sizes, produce less force compared to adults. Using the MATLAB function NMF, and for the NMF, we acquired the matrix coefficients that separate the raw EMG signal into the two control signals associated with extension and flexion. Sometimes it was necessary to invert the control signal so that from a right-handed perspective, extension corresponds with moving the cursor to the right, and flexion corresponds to moving the cursor to the left. It was reversed for left-handed subjects. So if they're using their left hand, extension would be moving the cursor to the left, and flexion would be moving the cursor to the right. In order to reduce unintended drift in the cursor movement, the subject was asked to resume a rest position with minimum EMG activity. A resting threshold roughly 50% higher than their average resting EMG amplitude was for each direction was established, so that basically any EMG signals that fell below that threshold would not contribute to the movement of the cursor. And because we're testing two independent variables, which is age and the gain level, a two-way mixed ANOVA test was conducted for each performance metric. In this table here, we define some of the useful metrics that we'll be examining, such as completion throughput, which is the time needed to complete the target acquisition, as well as first touch throughput and adjustment time. Okay. So moving on to the results. Here we have the first result involved the highly independent movements. We found that children had far fewer highly independent movements and higher variation than adults. We found a pretty significant correlation between age and the number of highly independent movements within children. However, we did not find a correlation in adults, as they all perform pretty similarly. And as you can see, the average number of hymns for adults was about 8.8, while children was about 5. So here we have the variance accounted for. The dimensionality of the forearm muscle EMG across 16 movements was estimated in able-bodied adults and children using the 8 electrode configuration. As the number of synergies used to reconstruct the raw data increased, more variance was explained by the NMF algorithm in both configurations. We found that the average number of minimum synergies for adults and children was 4. And using a 2-sample t-test, we found that there was no significant difference between these two groups. The quantification of the dimensionality of the EMG data across the broad range of movements are shown here. The muscle synergies can normally be visualized and compared across subjects or between limbs if precise anatomical electrode locations are used. However, this was challenging to do because in the present study, the given inconsistency of electrode placement was used. Therefore, we took a different approach to qualitatively demonstrate the underlying neuromuscular structure of the distracted synergies. Um, so here we see there's two assumptions. The extent to which a given muscle synergy is activated varies across different movements. And two, the subjects share similar muscle synergies that are associated with movements of one or a set of joints. These assumptions can be justified by previous studies that have examined muscle synergy in upper limbs. The synergies here were normalized to zero mean and unit standard deviation to remove scaling ambiguity of the NMF algorithm. So we can see that in both adults and children, the synergies seem to take four distinct directions, which can be thought of corresponding with flexion, extension, and abduction and adduction. So very quickly, the failure rates are shown here for both groups. The failure rate increased with gain level, and children having significantly higher number of failures per test. Although it's not recorded, it is suspected that children on average had higher levels of co-contractions, which could be quantified by taking the minimum magnitude between the left and right control signals. And co-contractions could also be the reason that children subjects on average had longer adjustment times, which is going to be shown later in this presentation. Figure 11 shows the completion throughput across all games over 6 tests between adults and children. We found that adults showed significant improvement in the last 3 trials compared to the second phase versus the first phase, while children did not show any improvement at all. So for that reason, we just, for the remaining metrics, we've only considered the data from the second half, as it provides a better representation of the expected performance. Figure 12 shows the average completion throughput across games, indicating the information transmission rate in order to complete the target acquisition. Here we observed a significant difference in both gain and age factors with p-values of 0.004 and 0.001 respectively, with no interactions between factors. Thus, we conducted a post hoc two-tailed paired t-test and found no significant difference between medium high gain for both groups. Comparing first touch throughput in figure 13, there's no significant difference between age groups. No significant difference between age groups was found, the p-value of 0.154, indicating that children have similar reaction speed to adults for each game. Additionally, two-tailed paired t-test between games showed that there was no significant difference between medium and high gains, but low was significantly different than both medium and high. The mean adjustment time between groups, shown in figure 14. For this metric, it was found that a marginally significant interaction between aim and gain level existed. Thus, 3 two-tailed paired t-tests were conducted for each group, low versus medium, medium versus high, and high versus low. Between children subjects, there was no significant difference between low and medium, while in adult in the adult group, there was no significant difference between medium and high gains. So a moderate linear relationship was found between the ages of between the age of children between the ages of 6 to 14 years old and the number of highly independent movements produced. This suggests that on average, children would require more training in order to produce more consistent EMG signals. We did not find any pattern in the classes of movements that were labeled as highly independent movements. This can be explained by the high inter-subject variation as suggested by other studies. We observed children performing more associated movements during the experiment than adults. Associated movements being defined as random movements that do not relate to the task at hand. So like moving around in their chairs or being distracted by looking around and other things like that. The fact that we saw no significant difference between the amount of synergies between age groups suggests that children can operate in the same level of degree of freedom as adults. However, this could be due to a limitation of the study and in a critique of the muscle synergy framework in general, in that one of the critiques of the muscle synergy framework is that it could be describing limitations or constraints of the task. Because in consideration of the attention span of the average child, we decided to only select 16 movements, while other studies have measured more than 40 different movements. So this could also be explained by this. So this difference in movement sets or investigated movement sets could explain why only 4 synergies was needed to account for the majority of the variance within the data between adults and children. And although the first touch throughput between adults and children was largely similar, children had significantly longer adjustment times compared to adults across all gains, leading to lower overall completion throughput. This could be due to higher levels of co-contraction, which is a condition where in which the muscles around a joint contract simultaneously to provide stability. So this is something that we saw a lot in children subjects. So to conclude, from our studies, we were able to quantify the EMG performance of children performing abstract tasks using 2 different myoelectric control methods and measuring several different metrics and comparing performance to adults. As hypothesized, children performed worse on several metrics such as lower number of highly independent movements and slower completion times. Other features and combinations can be further explored to compare performance. Children did perform similar to adults in regards to the dimensionality of the muscle contractions produced. However, the lack of learning observed suggests that children may have difficulty using simultaneous proportional control systems. Furthermore, further studies should examine if it is indeed possible for children to be trained to control prosthetic devices developed for adults. Overall, a low to medium sensitivity gain setting is suggested, as this resulted in the lowest number of failures and did not sacrifice on overall throughputs. For future work, it's suggested to examine the anatomical, to examine the synergies using anatomical positions of electrodes, as this would give a more definitive understanding of which muscles are contributing to these synergies. Another thing that could be suggested is using a high density electrode array. There's definitely a lot of different features and classifier combinations that could still be explored. One of the limitations of this study was that we focused on abstract tasks, which is not necessarily representative of how one would use these two control methods with an actual prosthetic device. So examining performance under functional tasks is something that could help provide more knowledge to understanding how children perform. And overall, a longitudinal study on training for children to see if they can be trained to use the prosthetic devices designed for adults. For example, there has been some studies, there have been mixed results with exergaming, which is a rehabilitation in which for amputees, which kind of like gamifies the whole process. And there have been some improvements noticed in children, but again, there's a large variability within subjects, age groups, which kind of results in mixed evaluations. And so here are my references, and that concludes my presentation.
Okay, thank you. So now I think it's time for anybody who is not in the committee to ask questions, if there's any. Anyone? No? Okay, so I think it's it's time for the question within the committee, I think. Let's see. I'm not sure if other people should exit at this point. Maybe. Okay, so let's just, Dr. Park, do you have any questions?
Yeah, I have a couple, but I'll probably just ask one question that kind of makes me more curious than others. By the way, it was very interesting work, and I think the methodologies and experiment all seem very solid. There's something that I may want to ask later. I wrote a couple down. One question that I had was, after seeing the plots, and you did mention in the conclusion that differences between child and adults in terms of their performance and even learning aspects could be induced from the level of concentration or cognitive function. How much, like, qualitatively, do you think that would have been coming to play? Because I read your conclusion and pretty much like overall thesis, and the experiment session was 60 minutes long, right?
Yes, exactly.
And were there any like breaks in between? Like, was there any, if you were to like redesign this experiment, what other, I guess, what changes or refinement that you want to make based on your observation and based on the data that you got?
Sure. Yeah. Definitely, time was one of the biggest constraints or factors that played a role in. I felt like in children's performance. From my observations, we saw that they did get distracted pretty easily. There were, like, again, there's a lot of inter-age variability. There were some kids who, they had really good focus. They were able to do it with no problem, but some kids, they were just it was very hard to have them keep focused. And so that's why one of the things that I was looking into was like, at least from that I've gathered from like actually interviewing the children during the process, a lot of them did play video games, and I believe that would have make the entire protocol was very simplistic and not very fun, I would say.
Yeah,
Um, there
not
was
entertaining much, right?
right.
If you incorporate some of the more visuals or more kind of game like aspects into this whole setup, then they might have probably shown more engagement.
Exactly. For the proportional control, there was one adult subject that suggested that you can make like a game where it's like you control like an airplane or something like that, and you have to move it to like a specific altitude, which would signify the target. And that would be, a lot more fun than just, simply moving a cursor around. But yeah, that would have taken more time, and I'm not a video game designer.
Was there? Go
Yes.
ahead.
Oh, for the first experiment, that's kind of more difficult because that's just basic repetition. So I know that was definitely a struggle. So it's kind of difficult to um, get ideas for how to make that more fun, or or maybe some sort of feedback has to be implemented so that the children can understand how they're doing in real time. I think that's what is the biggest factor, because during the experiment, they're just sitting there looking at the screen, and the only thing that they see is like the picture, and that's it. So there were times when children did not perform the movement when it was recording, because they were distracted. So there could be design changes to make it more fun, so that they provide more attention to the actual task, or there could be changes into how the data is recorded so that it only captures when they are actually performing the movement.
Mm-hmm. Yeah. Another thing that kind of was interesting to see is the children showed higher level of co-contraction. And I think your reasoning was because they were not really accustomed to this, like utilizing their muscles or kind of following the instructions. Or what was the main reasoning on seeing the higher level of co-contraction?
From what we saw, one of the possible explanations was because of due to EMG crosstalk, because they have smaller forearms, so the electrodes are placed much closer together. So that it could be a reason why we're seeing more co-contraction, simply because the EMGs have more are picking up more crosstalk compared to adults.
But we are talking about flexor and extensor, right?
Yes.
I mean, I get that, EMGs that are in close proximity would have some crosstalk, but co-contraction, we are comparing the level of magnitude between the flexor and extensor. Am I correct? I'm not that much familiar with the.
Yeah, that sounds correct. The other reasoning that could be that could explain why they would use more co-contraction is that they're not used to using their muscles like that in an abstract task. So it could so it's possible that the instructions weren't clear enough. so that it wasn't intuitive for them. I think in order for them to get the cursor to stop moving, they simply had to relax their forearm, which is definitely capable. Like any children is capable of doing that. However, even with instruction, like the children just it was just not something that they seemed to comprehend all that well, on average, at least.
Yeah.
And that's what led to the higher adjustments.
Yeah, I think that that's a good explanation. And I would love to see some of the video later, not right now, but it'll be good to see the experiment setup and so forth, because yeah, that would be interesting to see. But I'll stop here and I'll leave the others to also have a chance to ask questions.
Okay, so Dr. Choi, do you have any questions?
Yeah, that was a very interesting talk. And my one question is, what was the acclimatization time period for the children? Because the number of synergy can be altered by the how people get used to use their muscles. So for example, if there is a plenty of time where was given to the children to replicate all of those, the different, the wrist movement, and the number of synergy would be less, I guess. And so do you provide, the same amount of time for the adults and the children?
Yes. So the EMG data used for the synergy analysis was the same EMG data collected during the movement repetitions, and each repetition was 5 seconds.
5 seconds, okay.
Yes.
No, but okay. And my biggest concern is, the placement of the EMG as you described. You're not placed the EMG because muscle synergy is trying to identify, the number of muscle recruitments in order to reproduce the specific dynamic tasks. So in that case, the measuring EMG of each individual muscle would be detrimental to get accurate synergy analysis. So for example, the synergy is trying to find, which muscle group plays, the dominant, what number of, the muscle recruitments is needed to reproduce to produce the certain dynamic test, right? So and then of course in the wrist there are many, the redundant of muscles of, the wrist flexors and wrist extensors, and people wanted to find out how many muscle groups recruit to create, the certain motion. But without measuring the specific EMG, then I think that, the result may be a little bit biased. And can you explain more about that?
Yes, that's a very good point. And that's why we suggested that for future work, the anatomical positioning of the electrodes should be examined when comparing these muscle synergies. For the purpose of this study, the way that we measured the muscle synergies was more so to kind of get an overall estimation of the dimensionality, because that dimensionality of the EMG data can be used to like estimate the number of degrees of freedoms that are able to be controlled. And so and since we were comparing that, and since that was the overall goal of this study, was to compare and contrast between adults and children, see if that was able to be like how similar that was.
Okay, and I have one more question. So in the the test protocol, the test conditions. Can you describe more detail? What are the test conditions? It's just simply to ask people to wrist flexion, extension, or is there any another the movements?
Okay. Oh, so for the second experiment, yes. So yeah, only during the okay. There, yeah, it was only we only measured extension and flexion. At least that's how we calibrated it. We used extension and flexion because it was just a one-dimensional task. It could be adapted for 2D and 3D tasks, which control which would rely on more degrees of freedom. Um, that is an additional study that could be done in the future to see how children perform using those higher dimensional tasks. But no, for this study, it was just this one-dimensional target acquisition that was based off of their flexion and extension. And obviously, the more force that they use, the faster the cursor would move to the left or right. And the targets were they were varying distances between the left and right, but equally distributed.
Yeah, I think the more complex motion would be more the more suitable for synergy analysis, because just simply doing the flexion and extension, there are not much the contraction is going to be needed. But if you ask people to rotate the wrist and then grab something, then that requires a lot of contractions in order to recruit a lot of muscle, the redundant of muscles, and then that could be more suitable for the synergy analysis in the future.
Right. So I think you have some confusion. The EMG data that we used for the synergy analysis came from the first experimental protocol, which where the subjects were doing all the 16 different
movements, right?
Yes.
Okay.
There weren't. Initially we were planning on doing individual finger movements such as this, thumbs up, point, whatever. But we removed those for time sake. And we did for the adults, we had them do those experiments or those movements. And yeah, we found that those that definitely could have had a different result on the overall number of synergies produced. But again, we had time constraints that we had to consider for children. We didn't want them sitting for more than 1 hour was already pushing it for them.
Yeah. So maybe in the future, like, the more practical, the recruitments, dynamic tasks such as, for the prosthesis, the people usually struggle with grasping the different object, different strategies, like holding the mug cup or what else, like, using, the hand to grasp the more complex object. And then that could be one good indicator for analyzing, the synergy for, the prosthesis application in the future.
Right. Yeah, exactly. Using functional tasks versus these more abstract tasks is definitely something that should be examined in the future.
Okay, that's it. Great work.
All right. So, Dr. Huang, do you have any question?
No, I don't have any questions. I don't think I'm formally on the committee, so.
Oh, yeah, but I just want to check with you. So then, so I think it's time for the committee to have a small, short discussion. So I think everyone besides committee can exit the room. And Miguel, I don't know, can I put you in waiting room? How do I put you in waiting room?
I think so, yeah. I just want to say thanks to everyone, my committee members, and everyone that showed up here to my friends that came to watch me defend. So thank you guys.
How do I?
Well, first, Miguel, I think your study was really interesting and it was well done. So congrats. Q, about your question, I think you need to become the host. Currently, Miguel's the host, and I don't think you can put a host in the waiting room.
Oh, okay.
So I need to be host. Okay.
Yes. So anyway, all right. Good job.
All right. Thank you for everybody.
Okay, I just
Okay, I'm the host now. Ah, I see. So I can then put in